Hi, Andrew Wolf here. In this video I'm going to provide a brief introduction to hepatic physiology, and then in future videos I will talk about the actual function of the portal venous system, and then I'll talk about the inner workings of the hepatic lobules, which are the working units of the liver. Okay, so in this introductory video I want to talk about sort of three main classes of functions of the liver. And the first class of functions is the processing of nutrients. So some of you may have heard of the liver described as an accessory organ to the GI tract. Well, that's because of its role in the processing of nutrients. Now, I always, having worked with patients in hepatic failure, I sort of chuckle when I hear people call the liver a accessory organ to the gastrointestinal system, because the ramifications of a failing liver go way beyond the gastrointestinal system. And when we talk about the other two functions, you'll understand this as well. It's not an accessory organ. Now, an accessory organ would be like the spleen could be an accessory organ to the hematologic system, and I would be fine with the term accessory there, because you can live without a spleen. You can't live without the liver. Okay, so processing of nutrients. There are three main classes of nutrients. There are carbohydrates, and the liver is involved in processing carbohydrates, in sort of mediating the balance between glycogenesis and storage, and gluconeogenesis. Now, and this is also, be connected with glycogenolysis, right? This is the creation of glycogen. This is the breakdown of glycogen in order to make glucose. Now, this is all sort of mediated by insulin and glucagon. So if you think about when we are in a state where we are making glycogen, we have a lot of insulin and not very much glucagon. So when does this happen? Well, this happens right after a big meal, right? So you have a big meal, your portal vein is packed filled with carbohydrates, and the pancreas has also been stimulated by the presence of glucose in the GI tract to produce insulin, and glucagon production at the same time, glucagon excretion at the same time is being suppressed. So we have more insulin, less glucagon, and lots of glucose available to the liver. So the liver produces glycogen. Now, glycogen is perfectly suited for storage because it's insoluble. So it's an insoluble form of a simple carbohydrate, and so it is stored in the liver. It is very bad for transport in the bloodstream, because in order to be transported in the bloodstream, it's easiest to be transported in the bloodstream, I should say, if you have a soluble molecule. So what's a soluble molecule? Glucose. So glucose is for transport, glycogen is for storage. So when there is lots of insulin, the liver is in storage mode and it's producing glycogen for storage. When there is very little insulin, or the insulin glucagon balance is tipped in this direction, this would be sort of in fasting mode, then we are going to begin the process of glycogenolysis and gluconeogenesis, so we can start cranking out glucose to be transported in the bloodstream. Now, I just want to make the point here that the liver is not the only place in the body that can engage in glycogenesis or gluconeogenesis. This can also occur in the brain and the muscles and some other tissues as well. The kidney actually is a very important organ that's also involved. But the liver, has a very significant function here. Okay, so what other nutrients? Processing of nutrients. Now, the next, the other two big nutrients groups are obviously proteins and lipids. So the GI tract has digested and broken down proteins into amino acids. Now the liver sort of puts them back together into pieces, and some of these are functional proteins, and some of these are sort of pre-hormones and other pieces of proteins that are going to be built up further in other cells. So the liver sort of takes the raw product of amino acids and either makes it into a functional protein or makes it into a piece of a protein that's not functional yet but will be made functional in other organs. And so when we start talking about the functional proteins, we're actually moving into another area of what's sort of considered another area of hepatic function, and that is considered the synthetic function. And we'll talk about that more later. So there's some sort of blurring of boundaries here when we get to talking about the processing of proteins. With lipids, the liver has a very important role. Now, interestingly enough, all of the water-soluble nutrients, that includes carbohydrates and proteins, are absorbed into the bloodstream, into the portal system, and go directly to the liver. The lipids, on the other hand, are not absorbed into the bloodstream. They are actually carried they're actually packaged into chylomicrons, and they're sort of too big to get absorbed into the bloodstream, so they end up draining through the lymphatic system, and in the lymphatic system they drain into the thoracic duct. So they work their way up through the chest and drain into the thoracic duct up in the left subclavian vein. So that's how they get into the circulation, and then they make their way to the liver via the bloodstream. So once they get to the liver processes can actually sort of take these chylomicrons and break them down and make them into triglycerides and cholesterols. And then, so we have a difficulty circulating triglycerides through our bloodstream because they're insoluble. So the liver also has a significant role in producing lipoproteins that can reversibly bind with these triglycerides and transport them around the bloodstream, and that's how you get the very low density lipoproteins, and then the high density lipoproteins, and these are all involved in transport of triglycerides in various stages. And I'm not going to get into that discussion right now. And then also the liver is involved with the storage of some vitamins and minerals. Okay, so that is the first function of the liver, the processing of nutrients. So the second one is the synthetic function, and I'll just put in the third one here while I'm talking about it. So we have number one, processing of nutrients. Number two, synthetic function, synthesis. And number three would be protection and clearance. Okay, so next we're going to talk about the synthetic function. So follow me down to a fresh spot on the screen here, and we'll talk about the synthetic function of the liver. Now this is where things start to get very interesting, particularly when we start talking about the pathophysiology of the liver. When you start to have derangements in the synthetic function, it causes all sorts of systemic problems, because this is a very important function that's central to our functioning as an organism. Now remember, when we talked about the hematologic system, we talked about how the blood has a lot of proteins, and the proteins are very important to the function of the circulatory system, because they produce what's called oncotic pressure that enables blood to flow back into the capillaries, to get pulled back into the capillaries on the venous end. So albumin is very important to the function of the circulatory system, and it also has carrier roles and things like that. In the bloodstream, about 70% of all proteins are albumin. The next important group of proteins are coagulation factors. Now, not all coagulation factors are produced in the liver, but a number of very important ones are. And obviously, the most important of all is fibrinogen, which is produced in the liver. And then most of the vitamin K dependent factors are also produced in the liver, and that would, for example, that would include prothrombin, factor 7, factor 10, of course, another very sort of central factor, and factor 9, to name a few. So really, I would say, I haven't counted them, but I would say the majority of clotting factors are produced in the liver, and without the synthetic function of the liver, the blood cannot clot effectively. So it has the liver is central in synthesis of coagulation factors. Now, another group of important proteins would be transport and binding proteins. And just to give an example of some proteins that we have talked about before, when we talked about the hematologic system, with the iron cycle, we talked about the importance of a binding protein called apoferritin, which when it's bound with iron becomes ferritin, which allows for the storage of iron in the body. And then a similar one is transferrin, that allows for the transport of iron in the body. And then we have immunologic proteins, and that includes all of the complement proteins and many of the proteins in the kinin-kallikrein system. So if you can imagine, if you lose synthetic function of the liver, I'll make a slide talking about the liver function tests, and the way that we measure loss of synthetic function in the liver with patients with liver disease is we look at prothrombin time. And it's not that prothrombin is the only important um, protein that the liver makes. It's that it is a marker for loss of all of these. So if you have a patient that has with liver failure that has a significantly elevated INR, you can also expect that they're going to have impaired albumin, they're going to have issues with transport and binding proteins, and they're going to have loss of immunologic proteins. So they're going to be essentially immunosuppressed, right? So keep that in mind, that we don't measure PT in patients with hepatic failure just because we're concerned about their bleeding. We are looking at it as a marker for all of the synthetic function of the liver. Okay, so what else does the liver synthesize? Well, one other factor that it synthesizes is bile salts. And when I talk about bile salts, that's separate with the conjugation of bilirubin is really not part of the synthetic function, but the creation of bile salts is. And bile salts are emulsifiers that are produced in the hepatocytes and move down, actually, and actually part of the synthesis is assisted by the cholangiocytes, and the bile salts move through the bile canaliculi and into the bile ducts down into the gallbladder, and then are excreted into the small intestines in response to cholecystokinin. And the cholecystokinin obviously is a hormone that is secreted when there is high fat concentrations in the small bowel. And so the bile duct is secreting these bile salts, and they're fat emulsifiers, so they sort of break up big globules of fat into a suspension of fat, so that the fats can be more easily absorbed in the GI tract. Okay, so moving back up to the top here, so we've talked about two elements. We've talked about the processing of nutrients, including proteins, fats, and carbohydrates, and we've talked about synthetic function of the liver. Now, the third thing that I want to talk about is the role of the liver in protection and clearance. Now, we haven't talked about the portal vein yet, but I know most of you, when I talk about it, a lot of it's going to be review. The portal vein is the vein that's draining the entire GI system, including the colon and the small bowel and the stomach and the distal part of the esophagus, etc. So if you think about that, the GI tract is full, particularly the colon, is full of bacteria. So, and the gut is pretty good at keeping bacteria out of the blood, but it's not perfect. Every once in a while some slip through. So the liver has a very important role in preventing bacteremia from enteric bacteria. This is a picture of portal circulation here, portal vein going to the liver. So it has a very central role in the protection and clearance of bacteria. So here I'm going to so the first thing is the protection from enteric bacteria. And so how does it do this? Well, the portal vein drains through what are called sinusoids, and I'll talk about this when I get into talking about the lobules of the liver. And the sinusoids are just specialized capillaries of the liver. And these sinusoids are lined with Kupffer cells. And Kupffer cells are very active, potent macrophages. And in fact, there are many of them, such that they make up a significant, if you weigh the liver by cell, they make up a significant portion of these cells. They'd probably make up about 10% of all cells in the liver by weight. So these are a significant part of the liver. Okay, and then next we have the breakdown of toxins. And those of you who have been in clinical practice or taken pharmacology courses, you know that the liver plays a very central role in breaking in the metabolism of medications and other toxins in the liver. And it has various ways of doing this. It can oxidize them, it can cleave proteins and break them in half so they're no longer functional. And also, one of the toxins that is in our body are sort of amino acids themselves. Amino acids can be damaging to the body, so the liver sort of deals with free amino acids by running them through the urea cycle and breaking them down into urea, which is something that's not harmful until it gets into really high concentrations. But this is easily excreted in the via the kidney. And actually, one of the breakdown products of amino acids, and this can occur either in our cells or in the gut itself. We can have ammonia, develop high levels of ammonia, and the liver runs this through a urea cycle and breaks it down into urea as well. And ammonia at high levels can be toxic to the body as well. Now, interestingly enough, since one of the central ways that the body breaks down toxins is by oxidizing them, remember, oxidation via free radicals can damage and break down proteins. So obviously it can directly damage important proteins in the body, and it also can damage DNA. So the liver uses oxidation to break down toxins, but it also needs to protect itself from oxidation, and so it produces antioxidants. One of the most important ones in the body, and that's produced by the liver, is glutathione. So the production of antioxidants is another important protective function of the liver. And glutathione is really the major one. So if the liver loses its ability to produce glutathione, or its glutathione stores are broken down or worn down too much because of too much oxidation, then the body is open to damage from oxidative stress. And actually, this is the major pathophysiology of acetaminophen overdose. Really is due to a rapid depletion of glutathione stores, and the liver becomes very prone to oxidative damage, and that can start to spread to other areas of the body as well. Okay, so that brings me to the end of my introduction to hepatic physiology and the three major functions of nutrient processing, the synthetic function, and protective and clearance functions of the liver. Please join me in my next video. I will talk about the portal venous drainage system, and then in my third video about the liver lobule, I'll talk about the liver sinusoids and the hepatocytes, and we'll get right down into the function of each cell. And as always, please take a moment to provide feedback by giving a thumbs up, and I'm willing to answer questions if you want to put them in the comments, and you can click here if you want quick and easy access to my other GI tract videos. Thank you. I'll see you in later videos.